As a biologist, I would tell you that ultimately life doesn't exist if proteins aren't on some level interacting with other proteins. So whether it's catalyzing force in your muscles or replicating DNA, proteins have to interact with other proteins to carry out all of the cell functions that are necessary for life. If there's ever cell dysfunction, it's oftentimes in some way, shape, or form tied back to some sort of protein-protein interaction. That's both the origin of many disease states, but also the opportunity for therapeutic development.
Protein-protein interactions govern almost every biological process and hold the key to treating cancer, infectious diseases, and neurological disorders. Yet, with only 10,000 antibody-antigen structures in public databases, we're building tomorrow's medicines on yesterday's data. Today, Troy Lionberger, who is the Chief Business Officer at Alphabio, reveals how measuring millions of interactions simultaneously changes everything. By generating unprecedented quantities of high-quality data, they are accelerating the discovery of rare antibodies, engineering better protein therapeutics, and training AI models that predict what works before you ever touch the lab bench. Let's explore how. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Troy, to the Smart Biotech Scientist. It's good to have you on today.
Thanks, David. It's a pleasure to be here.
Troy, share something that you believe about biotherapeutic development that most people disagree with.
It's an interesting question. I think the most controversial view I harbor right now is, given my background, I think there is an overwhelming historical acceptance that antibody therapeutic development is artisanal and bespoke, that you're really hunting for needles in a haystack, if you will, is often a common analogy that people are describing. And I think the controversial statement I would make is that it's far more systematic today than I've ever imagined. For example, I think most people are surprised when I tell them that there are tractable and reproducible ways to make antibodies that have the same affinity for their human therapeutic target as animal targets. I mean, not just cross-reactive, but the same quantitative affinity, which could help streamline preclinical development, for example, of therapeutic antibodies. Most people that I've spoken with seem to think that that is flight of fancy. And I think fundamentally there are processes that make this happen today. I think that is the most surprising thing I share with people on a day-to-day basis.
And this will open avenues to novel therapeutics and also more efficacious drugs.
That's right. I mean, preclinical development of antibodies is fundamentally the constraints in developing these therapeutic molecules. In large part, getting them to work with animal models that you require in your studies is problematic. Oftentimes, the affinities of your molecules for those animal targets are far worse than for your human targets. So while you have a drug that might work, in fact, quite well in humans, can't ever get to clinic because the animal might have toxicity issues because you had to put so much drug into it.
Before we dive deeper into today's topic, take us back to the beginning. What first sparked your interest in biotech, and how did that journey lead you to Alphabio?
The origin for me was really in college when a faculty member teaching structural biology started describing proteins as nanomachines. I think that visual has always stayed with me. It got me interested in science, wanting to understand how it is that these fascinating machines that operate with very different materials and properties than anything we've created as human beings, how they function and how they work. I think that naturally led to understanding that these proteins that we barely understand ultimately are the origin of all human disease, which ultimately lead to cell dysfunction, which we describe as disease states. It's ultimately the understanding of those basic building blocks of life. That is the challenge of biotechnology, for us to understand how it is that these proteins ultimately can be manipulated and controlled in a way that can elicit therapeutic effect. So to answer your question about how I ended up at Alpha, my career in biotechnology started at a life science tools company called Berkeley Lights, where I helped invent a really exciting technology to discover therapeutic antibodies. And that naturally led to working with many teams in the industry to support their discovery efforts, and became ever more aware of the next constraints for them, which was the preclinical development of those drugs, which is the problem that we are in large part trying to solve at Alpha right now.
When I looked at your website, what stuck out to me that your company, Alphabio, describes itself as a protein-protein interaction company. Why are these interactions so fundamental to drug discovery, and why are they also so difficult to characterize at scale?
Yeah, it's a great question. My background is in biology. As a biologist, I would tell you that ultimately life doesn't exist if proteins aren't on some level interacting with other proteins. So whether it's catalyzing force in your muscles or replicating DNA, proteins have to interact with other proteins to carry out all of the cell functions that are necessary for life. If there's ever cell dysfunction, it's oftentimes in some way, shape, or form tied back to some sort of protein-protein interaction. That's both the origin of many disease states, but also the opportunity for therapeutic development. Being able to characterize these protein-protein interactions. There are many technologies that have come forth to help do this. Surface plasmon resonance is an industry standard way of measuring protein-protein interactions, and we call this affinity. So understanding the strength of those interactions or the affinity of those interactions is ultimately the way that biophysical characterization describes these protein-protein interactions. And the problem historically is that these technologies are quite, as advanced as they are, they're also quite costly and difficult to use. And when I say difficult, people do this every day in labs all around the world. So it's not difficult to do a few, but if your goal is to make millions and millions of those measurements, it's just not a scalable technology. And a great example is to generate the amount of affinity measurements that come off of just one of our experiments that takes a few weeks at A-alpha. The equivalent amount of affinity data would cost you between $1 and $500 million. So if you were to do this using surface plasmon resonance at a CRO, it would typically cost a few $100s. We do 1 million measurements at a time. There's the math. That is, I think, the fundamental constraint is the industry, despite the fact that there is increasing awareness and appreciation that getting this volume of data is really transformative in elucidating understanding about biology. No one's going to pay $100 million for a week's long experiment. So that is really the constraint that we're trying to solve in the industry, is to make these data that are otherwise far too expensive and too hard to generate easier, more affordable, and more economical.
That's exciting, and that's definitely the way to go, to be able to screen a lot more and then find quote-unquote needle in the haystack, right? But for a much smaller, modest budget. If we just look at the general picture, because drug discovery has been done for decades, how do companies today do this traditionally? What are the traditional workflows, the methods? Just that we start with the basics.
The branch of therapeutic discovery that I come from is called in vivo discovery. In vivo discovery, you are typically relying on an animal model whose competent immune system is ultimately responding to the presence of an antigen that's presented and raising an immune response against that antigen. And on the discovery side, scientists will access those antibody-producing cells, identify those cells that are producing an antibody that is very specific to your disease target, and then sequence that antibody to then move forward in developing it into a drug. There's a complementary approach called in vitro discovery, where you use what are literally called panning methods. You could imagine gold miners panning for gold, which gives you an appreciation for the basic philosophy behind current discovery, which is needles in a haystack, mining for gold. In panning and phage panning, you're using bacteriophage to express a version of your therapeutic and access large diversities. So many different combinations of different molecules you expose to your therapeutic targets, find those that binds, and again, sequence them and move them forward in your development process.
I imagine there are a lot of advantages in this traditional approach. Can you highlight what are the advantages and what are the limitations?
So the advantages of the in vivo approaches using animals is that you're taking advantage of really one of the world's most sophisticated ways of generating diverse sequences of antibodies, which is a competent immune system. To date, while there is promise in AI has not been able to generate diversity of functional antibodies that a competent immune system has. That is the promise in the future of where in silico methods are going. But to date, hands down, one of the finest ways of generating a diverse antibody response is using an immune system. The advantages to that are the diversity. The disadvantages are, in many cases, you're not able to get human antibodies because it would be unethical for us to immunize human beings for the purpose of generating antibodies for therapeutics. So we're limited to animal models that produce antibodies that have to then be further engineered to be compatible with human biologies. We have developed humanized animal models to solve that problem, but these are expensive and not commonplace. That is the challenge there. On the in vitro side, using phage panning, it's much faster. The downside is there are oftentimes biophysical characterization issues with those molecules. For example, we're phage panning at room temperature. Antibodies have to survive body temperature. If they melt and dissolve at body temperature, that's a problem. So there are other liabilities with the in vitro technologies.
And now with the new technologies advancing very rapidly, what is the picture you're seeing? Are we going to have a side-by-side or eventually AI, machine learning, and so on will take over?
It's definitely top of mind for me personally. And I should be upfront and say this is the first time I've worked on the machine learning and AI side of the industry. So I'm definitely new to the game. So with that caveat, I'll just say I've mentioned in vivo, in vitro, and now, as you mentioned, in silico approaches, which are now, I would say, complementing the first two antibody discovery approaches. So de novo antibody design is the name of the field that is essentially trying to predict sequences of antibodies that will bind efficaciously to a therapeutic target. Right now, I see all of these as complementing one another. There are, as I said, advantages to in vivo and in vitro technologies. In silico approaches are often taking the output of those approaches as inputs to their models. They're absolutely interlinked today. I think the promise of in silico methods are to eventually amass enough data that you can generalize these models so that you don't actually need a wet bench. But I would argue the constraint is always, even if you didn't need data to train your models, you will need data to validate your outputs. There's no escaping the data cycle in this space. I think there is a lot of talk in AI about AI models essentially taking the lead. I think there are a lot of advantages to de novo design in terms of epitope accessibility and future next-gen molecules being created. But as it stands today, I would describe them as complementary.
And I imagine that with AI and machine learning, you will be able to accelerate the workflows. It's not the one or the other as I'm hearing, but it's definitely making certain steps of the process faster and more efficient.
That's absolutely right. And I'll give you a great example of that, which is a field that Alpha is heavily involved in right now, which is optimizing antibodies to be lead candidate molecules in preclinical teams. What would typically happen after the discovery of an antibody is done, you would then want to optimize that candidate. And that could involve making it more human-like so that it doesn't interact negatively with the human immune system when it's injected into patients. It could be improving developability, like what the propensity is for aggregation, how much you can produce from cells at scale. But you're also optimizing affinities. Historically, this has been a very, very complicated process where you first focus on driving your affinities to where you want them, and then you worry about secondary characteristics of molecules that may make them more manufacturable downstream. And then once you make those changes, you have to go back to making sure that your affinity hasn't veered off course. So this is a slow, recursive, and iterative process that is expensive and time-consuming at each one of those different phases that I described, which oftentimes in series ping-pong back and forth through various parts of the value chain. And that could take upwards of 1+ years of hard work and a lot of money spent. What we're doing right now, leveraging a combination of, I think, disruptive data generation to inform machine learning models in a very bespoke way, we're able to generate data sets that could basically train a model to be predictive of higher affinity antibodies, where you can actually insert the number of mutations that are required to make this antibody more developable and higher affinity. We can insert up to 21 of those mutations and still be greater than 50% accurate that it will maintain higher affinity than the parental antibody. So that gives you a sense of the flexibility that's involved here, where there was a time where you'd be hard-pressed to be able to add 2 or 3 mutations to an antibody to design it to have these characteristics you're looking for. Now we can very predictably train a model so that we can insert 20 or more mutations. So you can make those molecules more human. You can predict sequences that are more developable, higher expression, higher affinity, higher stability, all at the same time in a 3-month process. So that's taking potentially years of effort and distilling it down to a 3-month end-to-end process.
So traditionally, drug discovery would take years, like 2 years, 3 years, or what is the benchmark? Before we continue, biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18+ months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, "David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs." Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode.
I think it really depends, and having been in this space as I have, it really depends on the approaches that are being taken and the hardness of a target. But order of magnitude, you could imagine the part of the process that I'm referring to, which is lead optimization, could be 1 to 3 years of time that we would be compressing to a 3-month process.
Wow, that's massive. Yeah, that's a big change.
Yeah, I will say after that 3-month process, we've also done things that can't be done typically in the historical process. So we are driving the affinities of preclinical animal models to the same to be as the human target affinity. That is really, I think, where things are getting pretty exciting. Asking the question, how does this change the dynamic in the overall ecosystem? If everyone's projects could essentially be accelerated through preclinical development because these molecules have been essentially tailor-made for the experiments they plan to do in preclinical development.
At the end of the day, we need to find effective antibodies. I mean, that's the purpose of the whole exercise. The purpose of the whole drug discovery process is to find efficacious or the most efficacious antibody. Now, what I'm hearing is we have technologies to accelerate the workflow. My question is, do these technologies also enable to find that very effective antibody, or do we need some other technologies on top of that?
No, I would argue that that problem that I just described is not just making a higher, more efficient process, but we are hitting target product profiles that are needed for these therapeutics. There is no sacrificing quality by accelerating the timeline, which is, I think, a rare example of that. In 3 months process, you actually starting to guarantee results. I mean, this is something that I never thought would be possible with a service provider.
Can you let us into how you're achieving that? What is the technology used? What are the major steps?
Yeah. The way that this works at a very high level is we will generate tens of thousands of mutations of a parental antibody. So we will randomly insert arbitrary point mutations. We will make mutants with 1, 2, or 3 different point mutations at a time. We'll then measure the affinity of those tens of thousands of antibodies against a panel of different targets. So not just the human target, but maybe the mouse and the cyno target, but also point mutations of targets or comparable family members or targets you want to avoid hitting. So early readouts of non-specific binding. So you get all of that data on not just binding, affinity, and cross-reactivity, but also specificity from each of those tens of thousands of molecules. We then use all of that data to fine-tune an AlphaBind model. A-alpha's computational platform is known as AlphaBinds. That AlphaBind model has been trained on close to 1 billion different affinity measurements that have been generated by the company. And that fine-tuning data from a very specific parental antibody will basically train that model to make predictions of mutations that could be introduced into the original molecule. And what the machine learning is doing behind the scenes is picking up things like synergistic and compensatory mutations that you may not, with your human eyes, be able to see any benefit of, but you can clearly see in the data. As a result of this fine-tuning model, we can be greater than 90% confident out to about 15 point mutations. It's a very high confidence of generating antibodies that are higher affinity. And really then we use off-the-shelf commercial models on developability to down select to also in parallel, I would say simultaneously to the affinity, we would be down selecting to developability metrics like expression, solubility, and melting temperature.
So you generate millions of affinity measurements in a week. And are you using yeast display and genomics to do that? Or what is the trick behind that?
Yes, just to get in beneath the hood a little bit. Our wet bench technology is called AlphaSeq. AlphaSeq is a yeast display technology where essentially we're taking advantage of yeast mating pathways in nature. In the wild, if you were to talk to a yeast geneticist, maternal and paternal, mom and dad, would be A and alpha. That's nomenclature from yeast genetics. So the A and alpha strains in the wild have these mating receptors that will ultimately engage and result in mating, so formation of a diploid cell. The diploid cell contains genetics of both maternal and paternal cells. And what our co-founder and CEO, David Younger, proved while he was a graduate student in David Baker's lab was that there's really nothing super specific about these mating receptors. You could indeed use any molecules with any measurable affinity, stick them on the outside of these cells, and if those molecules had some measured affinity, they would actually increase the rate of that diploid formation or yeast mating. What that all translates down to is if you have a cell culture with a library of different A strains that, for example, might express an antibody library, and another library in the same solution of alpha strains that are expressing targets that you're interested in understanding whether antibodies are sticking to or not. Long story short, you can let the cells sit there in culture under certain conditions the team has optimized, and after some period of time, sequence everything and the more you would see a gene counts, the higher the affinities. Again, the diploid cells contain the genetics of mom and dad. You have essentially a genomic barcode corresponding to antibody and antigen. You count the number of times that you see that in your genomic readout, and you can quantitatively relate that to binding affinity in molarity, which is the traditional unit. The correlation is to standard measurements that I mentioned, like SPR, surface plasmon resonance, or BLI, biolayer interferometry. The correlation is about 0.85, so it's not absolutely perfect, but it's certainly good enough for the applications that we are using it for. The simple trick, if you will, is to be able to extract a biophysical measurement from a genomic readout. Genomics just happens to have a tool in next-gen sequencing that we can use for accessing the scalable data that we're trying to access right here and relate it to affinities.
You have a good correlation, and I'm curious, what is the affinity range you're able to measure?
Great question. Affinity range we're usually seeing, and I'll caveat this by saying that there are ways we can tune our overall culture conditions, if you will, to tune it to a more or less sensitive range. But a typical experiment would be order of magnitude hundreds of picomolar all the way to tens of micromolar. So it's a very large dynamic range that covers the therapeutic range that most people would be interested in.
That wraps up part 1 of our conversation with Troy Lionberger on the data revolution in antibody discovery. We've explored the limitations of traditional methods and how Alphabio's AlphaSeq platform is changing the game. In part 2, we'll discover how machine learning transforms this massive data set into predictive power. If you're finding value in this episode, please leave us a review on Apple Podcasts or your favorite platform. It helps other scientists like you discover the show. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.